Thank you for having me. It's a pleasure to be here and to talk about both, just vaccines in general and our focus on developing what we call the next generation of DNA and RNA vaccines for COVID-19. Let's see, okay, see how advanced my slide here is. Hang on a sec. There we go. So I just have a couple of disclosures. I am a co-founder of Orlance. It's a biotech company that is aiming to commercialize a gene gun liver DNA RNA vaccines that I'm going to be talking about. But I have currently no financial interests with HDT Bio. It's another biotech company that is aiming to commercialize a vaccine called line replicating RNA CoV2S that was evaluated for safety and immunogenicity in my lab. And so just to kind of start out, just give you an idea of where is the current landscape of our leading vaccines that are now in phase 3 clinical trials. I think that we're getting close to about 11 vaccines, either at late stage in phase 2 or already in phase 3 clinical trials. This is just summarizing really the 4 leading ones that we're hearing about in the news all the time. Moderna and Pfizer have an RNA vaccine, and AstraZeneca and J&J are developing adenovirus-based vectored vaccines. And you can see from, their sort of timeline or expected timeline that they're going to be doing some interim analysis of their phase 3 trials. And that just basically means they're going to look at the number of infections and compare the placebos versus the vaccinated groups and see if there is overwhelming evidence that the vaccine is working. Then they may be applying for emergency use authorization. And if that is successful, they're in some of these groups like Moderna and Pfizer are anticipating a potential limited use of their vaccine at the end of this year. But the majority of them for mass vaccination, if all is successful, everything's safety, and they meet the benchmarks that have been established for efficacy of a minimum 50% in at least 2 months of immunity, then you can see potentially licensure. Some of these vaccines rolling out in Q2 and Q3 of 2021. So that's the expectation. And as we get closer to those timelines, we're becoming increasingly optimistic of the chance of these vaccines actually meeting that goal. So one of the things, of course, we've been thinking about is what might be the potential shortcomings of those particular vaccines. Their rollout is going to have, I think, a significant impact in helping to slow down the pandemic. But we all recognize that all vaccines are not perfect. And so when we think about really what a pandemic vaccine needs to achieve, we believe it really needs to induce durable immunity. It needs to induce it quickly, ideally not take months to induce that immune response, and also last at least 6 months. Right now, we don't know if any of the current vaccines in phase 3 clinical trials are going to be any more durable than 2 months. So this may be an issue with the vaccines that are currently being released. And my lab, as well as many other labs, are looking at alternative vaccine strategies that might be able to increase that durability. We also think the most effective vaccine against SARS-CoV-2 will be one that induces both antibody as well as T-cell responses. The majority of the vaccines that we're seeing in phase 3 trials are really focused on inducing neutralizing antibody against the spike protein, which, in animal models has shown to be very effective when these animals are challenged, when the antibody is at its peak titer, the animals are protected. So there's a lot of optimism of the role of antibody. But there's also emerging evidence that T-cell responses can play an important role, and particularly as antibody wanes, which is, it's well known for coronaviruses that antibody responses are not as persistent as, say, for other sorts of viruses, that the T-cell responses induced against the virus may play a critical role. In particular, CD8 positive T cells can identify infected cells and eliminate them, and CD4 positive T cells, we believe, are going to play an important role in the memory response and in a rapid recall of antibody to help control the infection. We also think an effective pandemic vaccine, and this is really critical, is going to have to work across different demographics. As we know, the elderly are among the most vulnerable, as well as immune compromised to SARS-CoV-2. And typically vaccines are less immunogenic in the elderly. And so we think it's really going to be important to develop vaccines that are going to be very effective in the elderly and meet a lot of these criteria. Finally, a lot of the vaccines, that are being developed I think they really meet the criteria for being very fast in terms of scale up. And there's all of the leading vaccines in phase 3 clinical trials are pretty well advanced in scaling up their doses. Whether they're cost effective or not is a question. But one of the things they all lack is that they are not stable at room temperature. All of the current vaccines that are in advanced stages of development all require refrigeration. Some of them actually require -80 freezer, and that's going to pose limitations for widespread distribution of the vaccine. So we think there's going to be a real need for other sorts of vaccines able to fill in these particular gaps. So we, at my lab, has been working on nucleic acid vaccines, that's both DNA as well as RNA vaccines, for nearly 30 years. And they've been around that long. They've been around since the early 1990s when some of the first DNA vaccine studies occurred. And right away from their earliest conception, it was recognized that nucleic acid vaccines offered a potential to be an effective rapid response in the event of a pandemic. And that's really primarily because it only requires the gene sequence of the pathogen to design. So as soon as the sequence of a virus has been identified, you can very quickly design a vaccine, a DNA vaccine or an RNA vaccine. Of course, the key differences between DNA is it requires nuclear delivery, so it needs delivery technologies to be able to efficiently administer that vaccine. RNA vaccines have gained a lot of attention in recent years because of some advances in terms of being able to deliver RNA vaccines into the cell, as well as improvements in formulation for their stability, because they were initially not very stable. So DNA vaccines sort of took the front run for a number of years. But the key to both DNA and RNA vaccines is not only that you can design them quickly, but they're very quick to scale up. You can deliver multiple antigens to potentially address viral evolution. A number of vaccines that are in phase 3 clinical trials for SARS-CoV-2 depend on an adenovirus-based vector, and these all run potential issues down the line of repeated dosing will result in the induction of anti-vector immune responses that could limit their long-term use. One of the important things about particularly DNA vaccines, and to some extent on RNA vaccines, I'll talk a little bit about that today, is that they are stable at room temperature. And importantly, they induce both antibody as well as T-cell responses. And so COVID-19 nucleic acid vaccines that are currently under development in my lab, there's a couple of platforms that we're working on. One is a replicating RNA vaccine that's delivered intramuscularly in collaboration with HDT BioCore that developed a novel formulation. I'm going to tell you more about that. We are also working on DNA and RNA vaccines that are efficiently delivered to the epidermis using a needle-free gene gun. And we're focused on a number of different sorts of immunogens, that are being developed in my lab. Of course, a full-length spike, which is what the majority of the vaccines are being focused on. But we're also looking at an RBD-focused vaccine and the conserved elements universal coronavirus vaccine, looking even farther into the future of whether we could design a coronavirus vaccine capable of inducing some level of immunity against future potential pandemics. And so the first one I'm going to really talk about is this replicating RNA vaccine. We did publish some of the data I'm going to talk about today in Science Translational Medicine in August of this year. And so we'll just kind of talk about that one and its full-length spike as an immunogen. And so just to tell you a little bit about self-amplifying replicating RNA vaccine, basically this is based on the Venezuelan encephalitis virus. It is called a self-amplifying or replicon RNA vaccine. And what that really occurs here is the transcription. With the replicating RNA vaccine, you first make the DNA. It's very easy to manipulate and manufacture and rapidly engineer. And then what's really different about replicating RNA vaccines in contrast to the messenger RNA vaccines like Moderna and Pfizer are developing is that it undergoes, it basically copies itself. It replicates, expresses a replicase that then copies the RNA, and in copying that RNA, it produces more vaccine antigen. And the idea of having more vaccine antigen means that this vaccine has potential to be more immunogenic. Coupled with that is really that this replicase in these viral proteins expressed by this vaccine also have inherent immunogenicity properties. So there's sort of a self-adjuvanting aspect to these self-amplifying RNA vaccines. So we envision this particular candidate vaccine as one that might overcome some of the limitations of other nucleic acid vaccines in terms of their immunogenicity and potentially achieving induction of strong immune responses in a single dose. There are some issues, though, that we had, we began to think about developing a replicating RNA vaccine. Is that the strategy at the time, you can either use a cell-based production, which incorporates the structural proteins of the virus, the VEEV, and that, of course, then results in the development of the vaccine that would actually be susceptible to this anti-vector immunity. What's more common now, of course, is formulating RNA with a lipid nanoparticle. Most of the companies that are developing messenger RNA vaccines are using lipid nanoparticles to achieve intracellular delivery of the RNA. There are some issues, though, associated with that, and that includes lipid toxicity, as well as as I mentioned, these particular formulations are, there's some degree of limitations in their manufacture in terms of requiring cholesterol, which is a bit of a shortage in short supply. But notably, this particular formulation requires refrigeration and at very cold temperatures to retain its stability. So what we try to address that particular issue is in collaboration with HDT Bio in Seattle is to investigate a lipid inorganic nanoparticle delivery formulation. And this is different in the sense that most of the lipid nanoparticles that we're familiar with actually formulate the RNA inside the lipid nanoparticle. This one differs in that the RNA actually goes on the outside. So, so what this consists of is a 15 nanomolar super paramagnetic oxide nanoparticles embedded in a hydrophobic oil phase. So there's already a long history of clinical use with this. And there's a squalene incorporated that's an adjuvant, and then the cationic lipid. And this cationic lipid forms this, the electrostatic association with the RNA that enables it to attach to this particular particle. And so one of the key advantages that we can see if we contrast lion formulation of RNA vaccines versus lipid nanoparticles is that with the lion, you get the RNA going on the nanoparticle surface. And so what you end up doing is you produce the lion formulation and the RNA, and you store them separately. And then you actually do a one-to-one mixing of them, at the bedside. So with lipid nanoparticles, the RNA has to be encapsulated into the lipid nanoparticle, and this is what actually is manufactured and then stored. And that's the difference here, is that then this requires a very special storage conditions, -80 freezing, whereas these two components can be stored separately at room temperature or just in 4 degrees Celsius, and they last for a long time. And so then you just do the one-to-one mixing process. They also can be scaled up very quickly separately, whereas a scale up in here will require a significant formulation step of the RNA into the lipid nanoparticle. And as I mentioned, lion doesn't depend on cholesterol, which is all the components associated with this formulation are readily sourced, whereas lipid nanoparticles, they do use cholesterol, and that is currently in short supply. And so we first, in collaboration with HDT, they first wanted to see whether or not the formulation with lion would be stable. And so what initially they looked at is when you formulate the lion with the replicating RNA vaccine, you can see that it forms an immediate complex formation with the RNA. And then when they tested at various temperatures, 4, 25, and 42, you can see that up to 25 degrees Celsius, it's stable for a number of weeks, even after it's formulated. Importantly, the lion it does actually protect the RNA from degradation. That's always an issue with RNA, that they can tend to be less stable. And even after the formulation, you've got the mixed formulation. Once lion and replicating RNA are mixed, at least for 2 weeks, is stable at room temperature. And so the next step was really to see whether or not this particular formulation was immunogenic. We first started our experiments in mice, and this just shows a simple scheme of what we did in terms of immunizing them on day 0, and then giving them a booster immunization, and then looking at different doses from 10 to 0.1 micrograms to determine an optimal dose. We looked at both antibody as well as T-cell responses in the lung and in the spleen. And so this just really shows our initial data here that you can see after single immunization, we're getting very high titered antibody that's boosted with a second dose, and the 10 and the 1 microgram doses induce comparable immune responses, but we see a fall off of those immune responses with the 0.1 microgram. Here we're showing that by pseudovirus assay or plaque reduction neutralization assay, we have that these antibody titers are neutralizing. And then one of the things that's really important for SARS-CoV-2 and a concern of all the vaccine developers is that the immune response that's induced is actually driven towards more of a Th1 rather than a Th2, since Th2 responses had been associated with SARS-CoV-1 years ago with enhanced pathology. And so here we evaluate Th1 versus Th2 by looking at the different antibody subtypes. IgG2C is indicative of a Th1 subtype, and you can see that the predominance of Th1 type antibodies indicates that the immune response is primarily Th1 biased. So we then looked at T-cell responses in both the spleen as well as the lung, and what you can see is that even at the very lowest doses, we're able to induce very robust T-cell responses with this vaccine in both the spleen as well as mucosal T-cell responses in the lung. And when we map those T-cell responses against different regions of the spike protein, what you can see is a majority of these T-cell responses are targeting the receptor binding domain. So then we wanted to determine whether or not this particular formulation might be effective in aged population. And we did this, modeled this in using aged mice. In this particular study, we compared 17-month-old, 8-month, and 2-month-old mice. Again, immunized them with a prime and booster immunization, looked at antibody as well as T-cell responses. And so immediately what we saw here is that when you compare across the different age groups here, and we're looking at both the 10 and the 1 microgram doses, you see no differences between the 8-month and 2-month-old mice. Then when you get to the 17-month-old mice, the lower dose, you do see a drop in the immunogenicity, but that can be rescued to some extent with the using the higher 10 microgram dose. And again, when we looked at the Th1 versus Th2 type subtypes, you can see importantly that in aged as well as in young, that these antibody responses that the vaccine is really inducing primarily a Th1 biased immune response in these mice. So what this kind of tells us is that most immune senescent populations may require either higher doses or potentially a booster immunization. And so one of the next thing that we looked at is whether or not we got good T-cell responses. And while we saw some drop in the antibody response at the 17-month-old animals, you can see here with the T-cell responses that we were able to induce with this vaccine robust and comparable T-cell responses irrespective of the age of the mice. And again, when we map those immune responses, we can see that the majority of the T-cell responses are targeting the receptor binding domain. And so having established good immunogenicity in a mouse model, we next wanted to do a pilot immunogenicity study in a non-human primate model as a preclinical study. As many of you are aware, non-human primates' immune system closely mimic that of humans, and doses that tend to work well in non-human primates usually translate well to use in humans. And so for this initial pilot study, we tested 2 doses, a 250 microgram dose, administering only a single immunization, or a prime and a boost immunization with a 50 microgram dose. We looked at antibody responses as well as T-cell responses. And so immediately what we saw in this study is we saw, importantly, no adverse events or local reactogenicity in any of these animals, indicating the vaccine safety. After a single immunization, you see the 250 microgram dose induced robust antibody responses, and those antibody persisted for quite some time. And then with the 50 microgram dose, it took a little bit longer for those antibodies to come up. With the booster immunization, we saw antibodies come up to the same levels that we see with the 250 microgram dose. Because these antibodies were on a trajectory of still going up, we don't know whether the boost was actually required to reach these levels of antibody in the 50 microgram dose. However, the prime and the boost with the 50 micrograms certainly equaled the prime only with the 250 microgram dose. We looked at neutralizing antibody by either pseudovirus assay or plaque reduction neutralization test. What you can see is that both of the doses induced significant and robust neutralizing antibody responses. Importantly, these antibody responses were on par with levels that you see in convalescent patients. So we're in the range of where a lot of the other vaccines that are well advanced in phase 3 clinical trials, these antibody titers are consistent with what we're seeing in those, and whether with a single immunization with a 250 microgram dose or with the prime and booster immunization. And so then we looked at the T-cell responses, and somewhat surprisingly in the non-human primates, in contrast to what we saw in the mice, you remember our ELISpot levels were in the thousands. Here what we're seeing is more of a modest T-cell response in the non-human primates. We saw all of them developed detectable levels of T-cell responses, but they were fairly modest, and in the ones that developed them, reasonable levels, they actually declined fairly quickly. So that's was somewhat surprising, but we saw similar results when we did intracellular cytokine staining, and we noted that we induced both CD4 as well as CD8 responses with the vaccine. Interestingly, when we analyzed the breadth of the T-cell response, it was quite consistent with what we saw in the mice, in that the majority of the T-cell responses were directed at the receptor binding domain. And so one of the things that, of course, I mentioned before is that we think an effective pandemic vaccine is really going to need to induce a durable antibody response. So we've been monitoring these animals for now a period of 6 months after an immunization. And what you can see here in this particular chart, we're looking at the same 250 and 50 microgram dose groups, is that the antibody responses after a single prime are still persisting for 6 months after that immunization. And the group that got the 50 microgram dose and one booster at 4 weeks, so their antibody responses are persisting, although one animal seems to be declining a little bit here. We then added additional cohort, went down a dose to only 25 micrograms. And interestingly, we waited to give this booster immunization until 20 weeks after the priming. And you can see that just with the 25 microgram dose, we were still able to induce high levels of antibody after that single dose. And by waiting longer to actually boost them later, we even see a further increase in those antibody responses. So this is actually good news from the perspective of wanting to achieve the goal of more durable immune responses. And so when we kind of summarize the key attributes of this, what we call the second generation lion replicating RNA vaccine, is that what we've seen is that it induced robust antibody and T-cell responses in a single dose. It was effective in aged animals. It has a long shelf life, potentially stable at room temperature. Its components are amenable to rapid scale up and are all accessible. And the immune responses, and so far in our animal models, are durable 6 months to date, and we're continuing to monitor them. So our next step is really to challenge these non-human primates. We did our studies in pigtail macaques and current non-human primate models of SARS-CoV-2 infection, have all to date used only rhesus and cytomologous macaques. So we had to first complete a pilot infection study in naive pigtails. And unfortunately, I don't have time to show those data, but they did show similar disease and infection to rhesus macaques. And so we just shipped our pigtails over to Rocky Mountain Labs at NIH, and they're going to be challenged next week. We have 3 cohorts. One group that will be challenged only 2 weeks after their final immunization, another 8 weeks, and another at 27 weeks post-final immunization. So, just kind of summarizing the development of this particular vaccine, we were able to, we started in January, immediately when the sequence for SARS-CoV-2 was published. We designed and produced and started testing this candidate replicon vaccine with only a few months. and did a preclinical down selection. This vaccine now is in GLP tox formulation and manufacturing, and is scheduled to go, if all goes well, into phase 1 human clinical trials in next month. And so we consider Lion really a first-in-class, second-generation SARS-CoV-2 vaccine, faster scale-up, higher potency with lower doses, more immunogenic in the elderly, and potential for durable immunity. And so I'm just going to turn our attention just for the last few slides to our DNA and RNA delivery by the needle-free gene gun. In these particular ones, we're going to be looking at the full-length spike as well as the an RBD-focused immunogen. And so just to kind of give you a little primer on what we mean by gene gun delivery DNA vaccines, is basically we take the DNA or RNA and we formulate them on microscopic gold particles, and then we use a gene gun as a delivery device that accelerates those gold particles in a stream of helium at supersonic speed, and those get micro-injected directly into cells in the skin. Numerous studies have shown that the penetration of these particles is very painless. We are now in developing a clinical version of this research device. But what I'm going to show you is data from our research device to date. This is actually a study we published a couple years ago with an influenza DNA vaccine in non-human primates, showing that gene gun immunization after only a single dose is able to induce protective levels of antibody, and after additional booster immunizations, very high titers. These are well exceed levels required for protection, and also notably robust gamma interferon T cell responses. So DNA vaccines are really well known for their ability to induce strong antibody as well as T cell responses. In that particular study, we did challenge those animals with a heterologous H1N1, and that study showed that we got a significant protection, and that protection was actually correlated with mucosal T cell responses localized in the lung. And so gene gun delivered DNA vaccines have been tested in human clinical trials, and they have consistently been shown to be very effective in inducing high levels of antibody after only a single immunization, 100% responder rate, strong CTL responses, protective levels of antibody. Notably, compared to other DNA vaccine delivery technologies, it requires 1,000-fold less DNA than other delivery technologies such as electroporation. So in these clinical trials are using only 1 to 4 micrograms of DNA. In contrast, other delivery technologies require 1 to 5 milligrams of DNA to achieve similar responses in humans. And so we're kind of begun to investigate gene gun delivered COVID-19 DNA vaccines, and we designed a number of different sort of vaccines or standard full-length spike that we've been talking about. Another one is our receptor binding domain focused vaccine. What we did was we took this receptor binding domain and we fused it to hepatitis core antigen. When that gets expressed as a DNA, it expresses as virus-like particles. And what you can see after a single immunization, we get really strong antibody responses, much stronger than just a whole spike. And of course, those go up with a booster immunization. We also compared that to, we're looking at a spike DNA replicon. That is a DNA vaccine that expresses our RNA replicon. So it's sort of a DNA vaccine launch replicon. And that sort of gave very variable results. I think we have some improvements to do on that one. And this is actually comparing it to the spike RNA replicon vaccine formulated with the lion. So you can see that the DNA vaccine is actually able to induce antibody responses on par with what we're seeing with our replicon RNA vaccine. And these responses may even be better after a booster immunization. So we're excited to see those and are moving those forward into non-human primate studies soon. One final note is that the gene gun can deliver RNA vaccines. It took us some time to figure out how to formulate and keep them stable on gold particles. One of the things, of course, with DNA vaccines is they work very well, but they have to get into the nucleus. So when you actually deliver the gold particles, DNA formulated in gold particles, not all the cells that get the gold particle are going to express that antigen because that DNA needs to get into the nucleus. And so we can get the DNA directly into the cell, but then it's just, passively taken up by the nucleus once it gets in there. In contrast, RNA only needs to get into the cytoplasm. So every cell that gets a gold particle will express an antigen. So we're sort of in the initial stages here, but we're excited to see this difference in what potentially gene gun delivery of RNA vaccines can do. And so just to kind of summarize and give sort of my acknowledgments here, I really want to bring credit to Megan O'Connor and Jesse Erasmus, two superstar postdocs in my laboratory, as well as my entire lab here, in collaboration with David Wiesler and Michael Gale at UW. Of course, all the monkey studies were done at the Washington National Primate Research Center. And then, of course, credit to HGT Bio for the formulation, NIH Rocky Mountain for, going to be challenging our animals, and of course, acknowledging my funding sources. And I'd be happy to take questions.
All right, Deborah, thank you very much for that beautiful talk. Amazing. So it's great to hear the next generation vaccines coming online. This will be great. I just have one question. So to what extent do you think people are worrying about replicons in general being an issue, that there's, persistence more than you want, and maybe reaction to the double-strand RNA issues more than you want some, but maybe more than you want, and or reaction to the replicase or other viral issues?
Yeah, that is something that we are definitely looking at. And I know that, Pfizer also has a replicating RNA vaccine that they're developing, and there's another replicating RNA vaccine that's coming out of London. And the main thing that we're looking at is really continuing to dose down. I think that's the key to sort of avoiding the potential high reactogenicity. I will note that our 250 microgram dose, which is about as high as we can go, we saw absolutely no reactogenicity associated with that in the non-human primates. But it's something that we're very much aware of. We're currently collaborating with Michael Gale here, who's an expert in systems biology, who's looking at the innate immune responses induced by the vaccine. So we want to hit a balance between, triggering just enough innate response to take advantage of that for increasing the immunogenicity, but not so much that we're going to get the reactogenicity. So we're very much aware of that. But we think ultimately it's going to be an advantage in the sense that we can get away with much, much lower doses of DNA. We haven't even found where our lowest threshold is. We're down to 25 micrograms in a non-human primate. We're going down to 5 micrograms in our next study.
Right. So those are huge advantages, and the potential durability is really important. Obviously, really interesting.
Mm-hmm. Thank you.
All right. So we'll see if there are any other questions from anybody. Let's see, we have maybe one here. Yeah, Larry Chasen asks, can you compare the price per dose for the lion RNA vaccine versus a protein vaccine?
Yeah, so I personally am not, able to give you a number, but the expectation is that a lion RNA vaccine is going to be much less expensive than a protein vaccine. That's just basically because of manufacture. With a recombinant protein vaccine, you're relying on cell culture, and there's a huge process associated with that, including the purification steps. In general, recombinant protein vaccines tend to be the most expensive. Live attenuated viral vector type vaccines would be less expensive, and nucleic acid vaccines are expected to be some of the least expensive.